So, last week we sort of just oriented everyone to the concept for this course, right, to the basic idea of what we're going to talk about. We kind of talked about the discipline a little bit, how this is physiology and behavior. We're going to talk about both things together, and just make sure everybody's starting out sort of at the same point. So we're all excited about that, right? I should have just said something, because I wouldn't have really bothered to think like I was sitting out there. I'll move this too. How's that? Is this in your way? No. And I can have a plumber come in and take care of that later.
Yeah, we might need that.
So we just sort of got oriented on things. This week we're actually going to get into the heart of some of the material and talk about some fun stuff. I will say, for those of you that have not read Chapter 2, which I'm going to assume is just about everyone, this is probably the most difficult chapter in the entire book, okay? So most people really struggle with this. If you read this chapter and you've decided you're going to drop this class, don't, right? Because it gets easier and then harder again, and then it gets easier again, and then one more time it's going to get harder. But then it gets a little bit easier right at the end, Trevor, and you're going to be fine. Okay, so just hang in there. I'm guessing most of you have to take this class, right? Anybody in here because they have to be? Yeah, that's what I thought. Okay, a lot of you are here because you have to take this. So hang in there. I'm the only person who teaches this on campus, so you have to struggle through it, right, Casey? But they can do it. People do it all the time. At least 33% of the people who take this class pass it. At least. I mean, sometimes it's more. It just depends. So, we're going to talk about structure and function of cells of the nervous system. For those of you who've never had a sort of physiology course, this is going to be a little more complicated than things maybe you've thought about in the past. For those of you that have had a physiology course, this might be just a little bit lighter than some of the things you've covered, or we might think about things in a slightly different way because, again, we're thinking about that behavioral side of things, right? So it's going to be a slight difference, but it's going to be fun, so hang in there. We will talk about different cells of the nervous system. We'll talk about communications within the neurons. We're going to think about inside of that cell itself, and then how that's going to communicate with other neurons, right? If you've got something really awesome to say, that's great, but if you don't share that with somebody else, it doesn't do a lot of good, right? How many of you have ever been out somewhere and all of a sudden there's like a guy in a bear suit with a knife, and you're like, well, that's something interesting. I should probably let everyone else I'm with know about that, and you decide not to. Bad things happen, right, Jacob? You don't do that. That's just not being a good friend. You see a guy in a bear suit with a knife, you let somebody know. It's just standard.
Dylan doesn't do that.
Oh, really? He looks like a guy that wouldn't do that. I don't blame you. Some people have too many friends. All right, so there's learning objectives. Don't worry about those. We really do want to divide the nervous system, though, and we're going to divide it in different ways as we go along. One way we're going to do this is with what's called the central nervous system and the peripheral nervous system. Now, most of you are thinking, I know this. This is pretty basic information, and it is, right? But we'll talk about why it's kind of important in just a moment. So the central nervous system includes your brain and your spinal cord, right? All that stuff right down the middle, it's definitely going to be encased in bone, right? Most people have a skull and that nice vertebral column, right? So those things are going to run in there. Anything that jumps off of that, right? Anything that jumps off of your brain or your spinal cord, it's going to be part of your peripheral nervous system. Okay? This includes things that actually attach to the brain, so we'll talk about cranial nerves and spinal nerves. Those are considered part of your peripheral nervous system as well, right? Even though you might think, well, they're directly connected, they're going to be part of the peripheral nervous system. Later, we'll figure out there are some important differences, sort of biochemically speaking, between the central and peripheral nervous system, and that is important for things like MS, right? So we'll talk about those applications later. In case you didn't understand what I was saying, here's a nice diagram. Hopefully none of you are colorblind. If you are, let me know in an email later, right? Because maybe I can make some changes to the diagram so you could actually see those. So the brain and the spinal cord there in kind of that nice, bubblegum red color. And then you've got your nerves and so forth out there in blue. Not too complicated. This is sort of a three-step process. If we start to think about, what is your nervous system really doing, right? It's really only doing three things. It's collecting inputs, right? Okay. So that's kind of step one. The second step is it's going to do some calculation, right? So whatever inputs you get, we're going to rumble those together in some kind of mathematical fashion. Don't worry too much about that. We'll talk about it as we need to. And then the third thing is that output. Okay? That's really all that your nervous system is doing. And Kyle, you can think about this from sort of the top down, right? So you can think about the whole organism is really just doing input, calculation, output, right? You're gathering information, you're processing that information, and then you're making some action based on that, right? But you can also take this all the way down, really, down to ion channels, right? Which are gathering some sort of input. Based on that input, they're making some sort of, decision about what they will do, and then they're making some kind of movement or shift in their shape. We'll talk more about that later. It's not making a lot of sense. And that creates some sort of output. So your entire nervous system can really be summed up into those three steps. And I think that's an important sort of three-step framework to think about as we go forward. Often they'll break it up into what they call sensory, motor, and interneurons. Right? We can apply that same three-step framework to this. You've got a sensory neuron. There's some guy wistfully staring at a glass of water. Like, wow, that must be the most tasteful water of all time. Really wish I had a much more in-depth relationship with that glass of water. Thinking about the past relationships with other glasses of water and maybe how those came up short. We're going to gather that info. We send it into that sort of interneurons, send it into somewhere in your brain. You don't need to know where that is now. We'll think about it later. You're going to do some calculations. How far away is that glass of water? Can I reach that just by moving my arm? Do I need to take some steps toward that glass of water? What are the other things I need to do? Once you've processed all that information, and this happens in milliseconds, right, folks? Much, much faster than I can describe that process to you. And then, Brandon, we create that output, and we're like, wow, bam, glass of water, right in my hand, right? And so you've created that output, whatever that is, that movement. In this case, we're doing it with a motor neuron. But you can take it and break it down into this, even if you look at just this inner neuron, right? It's receiving information from that sensory neuron, so it's gathering inputs. It's doing some internal calculations we'll talk about in a few moments. And then it is letting that output neuron, that motor neuron, know, hey, you need to create some kind of signal, generate some sort of movement, right? So wherever we decide to chop this system, we can still use this three-step framework. Here is your basic generic neuron, right? This is not drawn to scale. Okay, so I'm going to be clear about that. Not everybody tells you that. This is also not really any neuron you're ever going to find in your body, right? They're all shaped differently. They've got funky arrangements, right? The reason for that is they're all going to perform some kind of different task, right? They're going to be very specialized for that. So the structure, the shape can vary from one sort of neuron to another. And we'll go from there. But all neurons have sort of basic components, right? So I think if you understand the basic components, what they do, anytime we swap out those components for something else or we make some changes, then you'll definitely be able to understand what's going on there, okay? So first we probably want to start, because we're thinking about that three-step process, right? Let's think about the dendrites for a moment. Really sort of irritates me when people call them dendritic trees. And I know that's like a real small point, right? But dendrites is from the Greek word for tree. So if you say like dendritic tree, it's kind of like saying tree, which if you go around and you just yell tree all the time, people are going to laugh at you. So, don't do that. It's a little better if you say dendritic arbor, but then you're just trying to be pretentious. And I think that's a problem too. So we'll just call them dendrites, right? And we'll go from there. And they do look like tree branches, right? And that's awesome. These guys are the main receiving apparatus of your neuron, right? Depending on your neuron, wherever that is, on average, you're going to receive about 10,000 contacts onto your dendrites, right? So about 10,000 inputs. Some, again, depending on where we're located, some neurons will only receive one input, right? So when we talk about the structure of your retina later, Tyler, we're going to think about like a one-to-one relationship, right? When we talk about the cerebellum and some of the Purkinje cells in the cerebellum, they receive 100,000 inputs, okay? So, but on average, if we say 10,000 inputs, we're going to be pretty accurate, right? So about 10,000 different cells are contacting, or different inputs are contacting these guys out here on these dendrites, okay? Just all over the place. Now these contacts, you can think about them, I like to think about them as like yes and no votes, right? Like, is this an up or is this a down kind of vote? Thumbs up, thumbs down, right? And basically what we're doing is we are saying, hey, you should probably get a little closer to sending an electrical signal, or no, you need to get a little farther away from firing an action potential, right? And we'll talk later about how this works, but the basic idea is every one of these contacts is going to operate an ion channel, right, or a series of ion channels. And they'll open and close, and they'll either make the inside of the cell more positive, or they'll make it more negative, one of the two, okay? Now, I know that doesn't make a lot of sense to you now, right? It might make sense to some of you. If you guys had a physiology course, you guys talked about action potentials. If it doesn't make sense to you, just let it sit over there for a minute. We'll come right back to it, okay? Because we're going to, again, on this carousel, we're going to come back around, okay, Scotty? So just hang in there. Now, all of these inputs are going to get collected out here in the dendrites, okay? That's kind of cool. The dendrites will take those signals and they'll kind of funnel them down in here to the cell body, or the soma, okay? When you think about the soma, that's sort of what we think of when we think of a basic cell, right? Everything that you need to make a cell operate is inside. So you've got a nucleus, you've got your DNA, you've got your protein-making apparatus, all of those other fun, pieces, the endoplasmic reticulum, all that fun stuff. It's not going to be terribly important to us for this course, but it might be important for something else. Other things that are in there, tons of mitochondria, right? Okay, we're definitely going to need a ton of energy to operate our neurons, right? These guys are real energy sinks, okay? So they're constantly doing something. We'll talk later about what that something is, right? But for those of you that might know, we've got a thing called the sodium-potassium pump, right? And that guy's constantly burning up ATP, right? It's constantly using ATP, so we've constantly got to be making ATP, constantly have to be supplying these cells with the components to make ATP. The problem is they don't have a lot of their own sort of storage mechanisms, right? So a lot of your cells have the ability to store these components. They can store up energy. Neurons don't have that, right? They don't have that capability. They're very specialized, so we got rid of all that stuff, right? I like to think about these cells like cars, right? I mean, if you wanted if you were designing a vehicle that was going to carry rocks, right? What are some of the things that you might not put in that vehicle? Well, like a bunch of extra seats, right? I mean, if you're just hauling rocks, well, you need a big spot for rocks. You don't need a spot for people, right? So we're going to get rid of seats, put in some more rock space. If you're, driving around a soccer team, well, probably like, a truck bed doesn't make, well, maybe it makes a lot of sense to some people, but some people like seat belts. So maybe something with a lot of seats in it makes a lot of sense, right? Instead of like space to haul building materials, right? So you get rid of whatever you don't need. For these neurons, they got rid of their storage capacity, right? Because they're going to do a bunch of other fancy stuff, and they really need to focus on that. Later we'll talk about support cells. In particular, we'll talk about astrocytes. Don't worry about those just yet. But those guys are going to help supply the nutrients that these neurons need for energy, right? So it's going to be awesome. So lots of business going on in the cell body. It's going to make neurotransmitters. We'll talk about those later. All of the proteins that you need for receptors and so forth. All the fun stuff's going to happen here in the nucleus and in the rest of the cell body. Once you leave the cell body, as you go out, there's a spot here called the axon hillock. Okay. This is sort of the point of no return, right? If you are sending an electrical signal and you get to that axon hillock, and as soon as you get past that, I mean, it's like the top of the roller coaster, and they let you go, you're going down and you got to ride that thing to the end, right? So you might just scream the whole way. They're not stopping, right? Because it's not going to happen. Okay, you got to wait until it gets to the end. And that's what we're doing here. Once we get to this point and we go, we're sending an action potential, it's going to go all the way down that axon, right? All the way here to the terminals. Okay. So let's talk a little bit about this axon. The axon is basically just a long cable, right? Carries a signal from the cell body out to the terminals, right? It's going to carry an electrical signal. It's going to be wrapped for the most part. Now there are times where we have things that aren't wrapped, but it's going to mostly be wrapped with something called a myelin sheath. We'll talk more details about that later, what makes that myelin sheath. It's not a continuous wrapping. There are gaps, right? And these little gaps here are called nodes of Ranvier. Please don't call it nodes of Ranvier. I will fail you. Okay, you may have heard it that way. That's people who don't know what they're talking about. This guy was a French Canadian. So it's Ranvier. Trevor, a Canadian, taught me all about ion channel mechanisms, so that's why I know this. So, sort of a point of pride there. So we've got our nodes of Ranvier. These are the little gaps in between the myelin sheath. Later we'll talk about how that gives that action potential sort of a little bit of a boost, right, as it's traveling, so that's going to be exciting. We get out of the myelin sheath, out of the axon, and we get down here to what's called the terminals, or terminal buttons. Sometimes those really pretentious folks will write it terminal boutons. They didn't write anything else in French. I don't know why they wrote boutons, but they did. So we'll just call them terminals. You guys can call them whatever you want. It all means the same thing, right? So these terminals will then actually contact back on to some other dendrite, most likely. They may contact some other places. Sometimes they'll contact directly to cell bodies or even onto axons. That happens. And some of them will even contact back onto other terminals, right? So we can have some interesting arrangements. Again, it all depends on how we want the structure of this circuit, right? So we've got a ton of flexibility here, okay? I don't want you to memorize all of these specific situations. I just want you to know that depending on the context, we can have the wiring different, right, Malik? So it doesn't really matter. Whatever the situation calls for, our neuron can be set up that way, right? And different parts of the nervous system are going to need different kinds of wiring. So we'll just set it up that way from the beginning, no worries. Okay? Now, at the terminals, that's where we're going to dump out neurotransmitters. A lot of you have heard of neurotransmitters. How many of you have heard of dopamine? Yeah, that's kind of the basic one. Serotonin, right? Acetylcholine. These are some of the big guys, right? You've probably seen them on those, like Paxil commercials, right? You got the little circles floating from one place to another. That's a neurotransmitter. We'll talk about what those do later, but just know that they exist. So that's a lot for one slide, right? But there's a lot of info there, so just be ready to unpack that. Hey, look at that. We talked about all those things. That was fun. Here's that same neuron, but kind of plugged in, right, to sort of that network with other neurons. And this is just showing you all of those, or some of those main sort of wiring diagrams that I was talking to you about, right? So you can see all of the contacts coming in from other cells. Multiply this up until you hit 10,000. You can imagine why they didn't draw all of them, right? So then you end up with some situation that looks like this. It doesn't make any sense to you, right? So we left a few out. Here we are contacting dendrites. Here we are contacting the soma. Again, we said, those are all possibilities depending on what we want to do. What's really interesting is when you make a contact kind of right here at that axon hillock, right? Since this is the point of no return, putting like a nice big stop right there in case you needed to hit the brakes would be pretty great, right? So right before you go, well, I'm going to drop this letter in the mail. You guys still use the mail? I don't know if people still use the mail. About 5 years, I'm going to wonder if the post office even exists, but they charge you $8 a stamp. But that's a whole separate rant for another class. But imagine you're getting ready to hit the send button on that email, right? Okay. How many of you have your email set up to ask you if you really want to send to that email before you send it, right? Like Gmail will do that to me sometimes. Like, are you sure you want to send this? Because it sounds a little angry. I'm like, oh yeah, absolutely. I really wanted it to be worse, but toned it down, so hopefully the filter didn't catch it. But you send the email. If right before then, Autumn, you could hit the stop button and you think, okay, that's actually pretty good, right? So sometimes you'll put a nice little terminal right there, and it'll kind of be that stop button, just in case you would need it, right? So it's kind of a fun thing to have. All right, now, axons are super long, right? And when I say super long, I don't mean like, 53 feet, right? Because that doesn't make a lot of sense. But compared to most of your cells, axons are massive, right? Axons can travel from one part of your brain to another. They span distances of some considerable length, especially at that cellular level, okay? When we looked at that neuron, let's just draw one here. Here's a cell body with some dendrites coming off. and there's that axon. Down here at the terminals, let's say we need some dopamine down there. Okay? Because you want to release some dopamine onto some other cell. That's going to be awesome. How are we going to get that dopamine down there? Where are we making that dopamine? Well, all of the protein-making machinery is really up here in the soma, right? Not down here in the terminals where we need it. So we have to rely on something called axoplasmic transporters. So we've got these things called microtubules. Okay. They're going to run the length of this axon. Okay. Most people think of microtubules as giving some sort of structure to the cell, right? And they do that in this case. They provide that structure, right? But they also provide sort of a railway system for us to travel up and down, right? So we can take substances from the soma, send those down to the terminals. We can also take things that are at the terminals and send those back up to the cell body for recycling and processing and reuse, right? So we're going to send things down, we're going to use them, we're going to pull things back. We can recycle that. We have sort of two types of movement. We have what's called anterograde, which means we're going soma to terminal. We have retrograde, which means terminal back to the cell body. It's that simple. I'm not going to really hit you too hard on the process of that. It's kind of an exciting system if you're really interested in axoplasmic transport. I suggest that you explore that. Otherwise, you can just let it go. All right, here's sort of a zoomed-in view of the cell body. Again, we've got our nucleus, we've got our mitochondria. There are those microtubules getting ready to go down the axon. All that fun stuff out in the cytoplasm that you normally have. Not going to be terribly important to us to understand how these mechanisms work. We will talk a little bit in this class about DNA and protein structure, right? We'll talk about that toward the end of the semester. And it's important in neurological disorders, right? Most of your neurodegenerative disorders are the result of misfolded proteins. Why do your proteins misfold? Well, there are a number of reasons. Some of those are related to your DNA, sort of getting flipped around and some mistakes in that copy process. But we'll talk about that as we need to, but right now I don't think we need to focus a whole lot on that. And there you go for that fun, DNA, RNA, protein sort of central dogma. You guys familiar with the central dogma? Somebody is, right? Somebody's nodding up here. I appreciate that. Or you just internally singing a song.
I kind of got it.
You kind of got it, yeah. So, if you guys have taken like a cell biology course or something, they probably talked to you about central dogma. And hopefully they told you it was wrong. Right? Did they do that, Gentry? They didn't tell you it was wrong. Now I'm really upset. So it typically goes, well, I can't spell DNA. That's bad. DNA, RNA, protein, right? And so they kind of always thought, well, and this was from Francis Crick, by the way, right? So he was kind of an important guy. We talked briefly about his literal partner in crime last week when we talked about James Watson. The problem with this is, well, you don't even really need DNA, right? So probably it was just RNA first, and then RNA to protein, and then later RNA was like, oh man, you know what, if I had like something to back me up like DNA, that would be sweet. And then DNA came around later. So central dogma is probably not so right, so don't get that tattooed anywhere. They've already started. I mean, maybe you could change it to like Dinah or something. Meet somebody named Dinah and they'll be your best friend. All right, supporting cells, right? We need to talk about those. Neurons are awesome, but as we've already said, they can't do the whole show on their own, right? Okay, they can't get the support that they need. Later, we're going to talk about something called the blood-brain barrier, which is super awesome, right? But some of you figure out interesting ways to bypass, right? Like on Thursdays, is Thursday still the day? Is that the blood-brain barrier bypass day? Nobody knows. It is. Gentry knows, right? That's the day. You don't have to tell me why you know. That was from that class you took, right? So we'll talk about that. So that's going to be important because they're not really great at protecting themselves either. Okay, so we're going to have some help with that. Sort of one of the first cells we're going to talk about are astrocytes. These guys play a bunch of awesome roles. They will suck nutrients out of the blood, pass that over to the neurons so that they can turn that into ATP. That's going to be pretty sweet, right? Because you're not able to get glucose kind of through that blood-brain barrier without these guys. We also have oligodendrocytes. These guys make that myelin sheath. Okay, they've got sort of a cousin called the Schwann cells that are in the peripheral nervous system. Perform a similar role, but they are biochemically different, right? And so that's going to be important for, again, some resistance to MS that we talk about later. And finally, we're going to think about microglia, right? These guys are responsible for the immune response in your brain. Anybody ever had a brain infection? You don't want one of those. You think it's bad getting some other kind of infection. You get one in your brain, and it's a real problem. So try to avoid that, right? Avoid using like water straight from the Ohio River in your neti pots. That happens to people sometimes. I don't know. People don't think. Why do we call these guys glia cells? Glia comes from the word for glue. It was originally thought that these glia cells actually hold your brain together. And while they don't necessarily do that as literally as people originally thought, they still sort of metaphorically hold things together, right? Because they still provide that support for the neurons that you have that are doing those important calculations. All right. So let's talk about astrocytes briefly. Why are they called astrocytes? Well, they're shaped like a star. Okay, they got very, very creative. They're like, wow, that's a star-shaped cell. Let's find a couple fancy words that mean star-shaped cell. Put that together and we call it astrocytes. If you know like some Latin or Greek, a lot of this becomes a lot easier, right? Because you start like, oh, astrocytes, that must be cells that are star-shaped. Just kind of think about that. So what do these guys do? One, again, they pull glucose out of the blood, right? That's where some people put glucose. That's a handy place to keep it for a brief period of time, so that your astrocytes can get their hands on it. They'll pull that out, and then, of course, supply that to your neurons, again, so they can do fun things with that. I don't want to give anybody flashbacks, but I have to say Krebs cycle just once, right? If it kind of hits you hard, you know what I'm talking about. You probably had to memorize that whole diagram, and you wake up in the middle of the night. It's on the inside of your eyelids. So you can have fun with that. I got a Nobel Prize for that. That was in student torture. That was the category. This was the category for that Nobel Prize. Student torture. So we're going to pull that glucose out. Again, structure of the capillaries in your brain is a bit different than the structure of capillaries anywhere else in your body, right? And we call this sort of collectively the blood-brain barrier, right? The point of a capillary really anywhere else is to be sort of a loading ramp, right? To allow nutrients and oxygen and so forth to fall out and like waste to kind of come back in, right? And so it's kind of this interchange, right? So we want to dump off things that we're trying to deliver to tissues, and then we want to make pickups, right? We want to kind of pick up the trash and move on and deliver that somewhere else later. And that's phenomenal, right? If you're delivering things to your liver, your kidneys, wherever, right? But your brain, given the delicate balance, right, in your brain, and that, well, your brain sort of controls everything else in your body, it might make sense for us to be very, very careful about what we allow into the brain, right? And so we might think, might be a good idea if we had some different setup here, right? And so we do, thankfully, have a slightly different setup of the capillaries in the brain. They're kind of tighter, right? Okay, so they're kind of wedged in there, tighter. Stuff's not going to leak out, okay? And we're going to have to, for the most part, we're going to have to pull things out through those astrocytes. Now, there are times, right, where we ingest things, right, either because we should or sometimes because we shouldn't, that bypass this blood-brain barrier, right? And I think one of the most important concepts in understanding life in general is what I like to call lipid solubility, right? It's just awesome. It's what I call a key to transcendental enlightenment. Once you get a handle on lipid solubility, the rest of your life makes sense, right? You know, stocks and bonds are nothing anymore, right? You guys are going to be like Wall Street wizards. You're going to be able to design, you know, cars that run on garbage. You're going to be able to like solve all the problems in your life as soon as you understand lipid solubility. All we mean by this is how easy is it for this substance to slide through lipids, right? And why do we even care if it can slide through lipids? Well, hey, guess what you're made of? Well, I have a bunch of lipids, right? All of your cell membranes are a phospholipid bilayer, right? So you've got sort of two sets of lipids, kind of there in a special arrangement we'll talk about later. If something is lipid soluble, it can slide in and out of there quite easily, right? So if you ingest something and it has some sort of what we would call a psychotropic effect, right? We really need it to get through there, right? So if you're on an SSRI, for example, right, a selective serotonin reuptake inhibitor, primarily prescribed for things like irritability, depression, lots of other things. It's a fairly commonly prescribed drug. Those would do you absolutely no good if they weren't lipid soluble. The reason for that is they would get in your capillaries, come through your brain, and there's no way they would be able to get out of that capillary, right? They wouldn't be able to get through that blood-brain barrier. But because they're fairly lipid soluble, they'll slide right out of there and they'll go into these neurons and they'll make stuff happen, right? There's another substance that's really lipid soluble, right? And this is for your Thursday nights, right? Alcohol is very lipid soluble, okay? Slide right in and right out of your cells, okay? That's why when you ingest alcohol, it does things. It like makes you think you're a professional athlete and you can leap long distances, right? It's kind of one of those inverse relationships. Like, here's what I think happens to my athletic prowess when I drink. Here's what actually happens to it, right? If you keep that in mind, you'll stay safe. Some people forget. Alcohol does something to memory too, so it's built in. So, lipid solubility. Now, there are two places, well, not two, there are a few places. We'll talk about a few of them. But there's one place in particular we want to think about in your brain where the blood-brain barrier is sort of weak, right? It's called the area postrema. Okay. That's the in the back, area in the back, you know, posterior. This is really great for projectile vomiting. Wow, who loves that? You know who loves it? Someone who just ate something poisonous. That's who loves projectile vomiting, right? Yeah. Oh, man. In case you think the world would be different if Joffrey Baratheon had an active area postrema instead of just like holding on to all of that.
I think a lot of people would be super dissatisfied.
Super dissatisfied. I think that kid could get a job now. He might still have a job.
Maybe he's just like, he doesn't want to act at all.
I don't blame him. I bet people spit on him in the streets.
It's not because he wants it. It's not because of that. He actually just wants to be a academic.
Just cash his checks. Yeah, that happens.
Especially like the nicest dude.
Yeah.
People say about him.
I don't believe that. So,
Just a punch in the face.
this will trigger, it's also known as the chemical trigger zone. So it'll trigger projectile vomiting. It's just which is good, right? If you do ingest something that you probably shouldn't, and it gets around to your area postrema, you go, whoa, I'm going to vomit that out right now. So that's awesome, so it doesn't cause more damage, right? So that's pretty cool. So it's right there for you, nice and safe. Eat all the poisonous stuff you want. This stuff will kick in eventually. Or not, one of the two. All right, let's talk about those oligodendrocytes a bit, right? These are fun. They're kind of like an octopus, right? So they're going to reach out and they're just going to wrap themselves around multiple axons, and they're going to reach out and wrap up multiple parts of those multiple axons, right? So one oligodendrocyte might make a handful of segments of myelin sheath on 3 or 4 axons, right? So that's kind of a good kind of thing to keep in mind. So if you lose this oligodendrocyte, right, you end up losing several chunks of the myelin sheath, right? Okay, you kind of lose out on that, which is bad. The myelin sheath does a very, very important job, right? It maintains the fidelity of that signal, okay? So it prevents that signal from leaking, from dissipating. Basically, it prevents a short circuit, okay? Prevents you from losing that signal, okay? So it's one of those, it's kind of like it keeps your internet working, right? So if you hit the send button on that email, that's awesome. You think, well, it left my computer. What happens is right as you're hitting send, your internet goes out, right? That email didn't go anywhere. And it was an important email, like an excused absence for a class, right, for an exam. And then all of a sudden you get a 0 on that because you didn't come to class. Your oligodendrocytes will prevent that from happening, right? Because they're going to keep those axons wrapped. They're going to make sure that signal goes from soma to terminals, and that's going to happen successfully, okay? If someone has multiple sclerosis, right, MS, the interesting thing about MS is it specifically targets oligodendrocytes, right? So it will target these oligodendrocytes, they will die, and you'll lose these segments of the myelin sheath. Okay? So folks with MS have difficulty getting their signals from the soma down to the terminals, right? So they have trouble sending those signals. So a lot of the symptoms that you see with MS, some of them are motor symptoms, so they have difficulty kind of generating those motor commands. Often they'll have difficulties with communication, right? Sometimes they have sort of delays, right, in their speech and their thinking because they're having trouble, again, sending those signals. A lot of it depends on the location of the loss of oligodendrocytes, but that's the basic idea. So, this is showing you with a Schwann cell. Not a big deal, but it's the same process. It's going to reach out, it's going to start wrapping around, and then it kind of creates that, nice little, jelly roll or something there for you around the axon. Now, there's an important difference between oligodendrocytes and Schwann cells. Actually, there are a number of important differences. One, at the biochemical level, they're different. Okay, so MS does not target Schwann cells. You don't see peripheral loss of the myelin sheath. With MS, you only see central loss. Okay. The second thing is a Schwann cell will only reach out and wrap an individual axon and create one segment. So if you lose a Schwann cell, you're only losing one segment on one axon, right? Instead of multiple segments on multiple axons, you're losing one. The other cool thing about a Schwann cell is if there is peripheral nerve damage, often the Schwann cells can help that axon regrow and redirect, right? This is why you'll sometimes see some peripheral nerve recovery if there's damage. You're not going to see central recovery, right? If there's damage to your central nervous system, you're not going to see that kind of natural recovery. If there's damage to the peripheral nervous system, you can see recovery. These Schwann cells will work together, and they will start to kind of make like a cone, right? So that they can kind of drive that axon as it regrows if it's been damaged and get it going back where it belongs. Not a perfect system, but it's much better than what you see in the central nervous system. All right, there's that blood-brain barrier. There's that area of postrema. Semi-permeable, like we said, some things can get through if they're lipid-soluble. Okay. Things that are lipid-soluble typically do not have a charge, right? So that means they're not going to have no charge. Or they could be really small. Even if they do have a charge, if they're super small, sometimes they can get through. Things that can't get through are things like ions, right? We'll talk a lot about ions as we go forward. Things with a charge aren't going to be able to get through very well. So we're going to kind of create that barrier. There you go. There's a capillary in most places. You can see it's got these nice big gaps here between the cells. That's super awesome because it allows things to flow in and out, which is really sort of the point of the capillaries. In the brain, we're going to seal those up. Look how tight those are. We got those nice tight gaps there, so nothing leaks out. Without the help, I'm going to draw an astrocyte there, right? Without the help of those astrocytes pulling things out. All right, somebody will spend a long time telling you about multipolar, unipolar, and bipolar neurons, but that somebody's not me, right? So I don't feel like this is really that important of a thing to spend a lot of time on, because there are people who are like spend forever like getting you to like, oh well, it's got multiple poles, well, and then it's bipolar or it's unipolar, whatever. I think the important thing to take away from this, Dylan, is we're going to structure that neuron however it needs to be structured to do its job, right? If a bipolar neuron makes sense, well, guess what? It's a bipolar neuron. If we want a unipolar neuron, it's going to be a unipolar neuron, right? Whatever shape and structure of that neuron that is necessary to complete that task, that's what's going to be there, right? So don't spend a whole lot of time trying to wrap your heads around this. I don't think that's terribly important. There are a lot of people who will just think it's really important to learn all of these classifications. And I think, well, that's fine. Somebody wrote a book on that. There's probably a chart you can download and you can put that on your wall if you want, right? What's important to me is that you kind of remember these concepts, right? And learn the concepts. Well, why would I even want a bipolar neuron? Well, if you're trying to get to two different places or you've got some other kind of interesting structure, bipolar neuron makes sense. We want to put that cell body not at one of the ends, but somewhere in the middle. Try to protect that cell body. We're gathering information here, sending information. Bipolar neuron makes a lot of sense. Okay? I'm not going to spend a lot of time on reflexes, right? Although this is the approved way to check if your iron is ready to use, to iron your clothes. I didn't know if you knew that. It's always the index finger on your dominant hand. Okay? Just keep that in mind next time you guys iron your clothes, or maybe the first time you iron your clothes. If you've ever used an iron, we'll figure it out. Here's that same guy. I mean, he needs a glass of water way over here. Just kind of. I don't know. They pick the worst faces, I think. I don't know who draws these. All right, so reflexes, I mean, it still sort of has that same basic input, calculation, output process, right? We could apply that there if we want. Not too big of a deal. Some reflexes have some sort of inhibitory neuron there. If you want to prevent that reflex from occurring. Some reflexes you can do this, some you can't, right? If you whack somebody in the knee, they're going to kick you in the face. That's a guarantee. There's no real way to prevent that. If you grab something hot, you can prevent from throwing a bowl of soup, all over the place if that's what you choose to do. Or just throw the soup. That's always fun. All right. I want everybody to take 37 seconds and think about your favorite cartoon. Gentry, I was being serious. Kyle, was it Tom and Jerry? I thought so. I'm that good. So my birthday was a few weeks ago, right? My nephew got me a card. I didn't even open the card. I just rubbed it and I said, "Is there a chimpanzee on this card?" Opened it up. There's a chimpanzee on it. It was impressive. I guess my Christmas gift from my mom last year, just because my wife told me how big the box was. She was like, "Oh, it's this big." I was like, "Here's what it is." It's exactly right. Okay, so the reason I wanted you to think about your favorite cartoon for 37 seconds was because we're getting ready to jump into something new and difficult and abstract. I just wanted to give you a mental break there for a moment, right? Think about an anvil falling on a rabbit or something. I don't know, that always helps. So, you guys kind of know what a neuron is, right? You know its basic parts. You know sort of what it does. It collects some information of some kind, it sends that information somewhere else for somebody else to deal with, right? What we want to think about now is the inside of that neuron. We know that the inside of that neuron is going to have an electrical signal go through it, right? And we want to think about the nature of that electrical signal. Right? And this is where it gets a little tricky, right? And what we're going to measure are what we call electrical potentials. Okay? And I think sometimes this can be a very sort of foreign and abstract concept, right? Like what is a membrane potential, right? So don't worry if it doesn't make sense to you. The first go around, we're going to hit it a couple times and hopefully it sinks in, right? Basically what we want to do is we want to look at that electrical charge across the cell membrane, right? So we're going to have the inside of the cell have a different electrical charge than the outside of the cell. And that's going to be awesome. Later what we can do is we can open up those ion channels, right? And we can allow ions to either move in or out, depending on what we need to do, and it can change that electrical potential on the inside, right? So, for simplicity, someone decided, and I think Brandon, this was brilliant, they decided the outside of the cell was going to be 0 millivolts. Okay? Just standard, the outside is 0. Okay? So what we do is we measure the inside relative to the outside. Okay? We've set the outside at 0. That means the inside is somewhere around negative 65 millivolts. Some books will say negative 70. I've even seen as high as negative 80. And it varies from cell to cell and brain region to brain region, right? Some are going to be a little different. Don't get caught up out of in the specifics, right? Just know negative 65 is where we're going to hang out. Okay? If your neuron is doing nothing, okay, it's not doing anything, just sitting there hanging out, it's going to be at negative 65 millivolts. Now, in reality, that never happens. Okay? Do you remember that time I told you it gets 10,000 inputs? Okay? What are the odds that all 10,000 of those inputs are like, well, not right now. That's not going to happen, right? Now, all 10,000 of them aren't sending a signal at once, but they're all sending signals at different times, right? So this cell is constantly getting some sort of input, right? Some sort of up or down sort of movement, okay? It's always getting a positive or a negative, an excitatory or an inhibitory sort of contact, right? One of those 10,000 is going to be doing something. So your cell's never exactly at negative 65, right? It's going to be hanging around negative 65, up or down a little, just as it's cruising along, okay? So how do we measure this? Well, what we do is something, we use something called a microelectrode, right? So basically this is like a super small thin piece of metal, and we're going to jab it somehow into the cell. We can talk about fancy ways to do that later. And then we need to put one obviously out in the outside of the cell, right, wherever that is. Now, mostly people are just bags of water. Okay? So really, like the next time you think like, well, man, I'm awesome, just think, no, you're just a big bag of water. So great job on that. You know, it's really sort of humbling, I think. The outside of the cell is primarily salt water. So really we're big bags of salt water. That's even better. You know. So when we think about that, the outside of the cell is just going to be a bunch of salt water, but it's going to be at 0, right? 0 charge. No charge there. The inside is going to be negative. If we want to fire an action potential, if we want to send information from one cell to another, right? We have to fire that action potential. We'll talk about that later, what that is. But what we need to do first is what's called a depolarization. Okay? This is when we are going to reduce the membrane potential, but we're going to make it go towards 0, because we're already negative 65, right? If we're reducing that, right, then we're moving to negative 60. Negative 55. Those are depolarizations, right? Those are moves in the positive direction, towards 0. Okay? If we move away from 0, farther away, that's called a hyperpolarization. That's when we become more negative. If we go from negative 65 to negative 75 to negative 85, right? We're hyperpolarizing. We're becoming more polarized. We're becoming farther apart, farther away from 0. Okay? Does that make sense to everybody? Okay. All right, let's talk for a moment about diffusion. You guys know diffusion. A lot of this is going to be based on concentration gradients, right? So this is kind of help setting us up for what's going to happen later when we're going to do some special things with ion channels. Now, if we think about our cell, we have the outside and we have the inside. Now, I told you outside the cell is mostly salt water, right? And so what's in salt water? Sodium and chloride. Both of these guys are ions, and we're going to have high concentrations of them, right? High concentration. So lots of sodium, lots of chloride. Essentially, we've got an unlimited supply of sodium and chloride outside of the cell, right? Because it's just a big bag of salt water, okay? So whenever we need some sodium, we need some chloride, it's there for us, we don't really have to worry about it, okay? We just open up a channel, stuff happens. So, inside the cell, we have potassium, that's a fun one, right? And we have something called an organic anion, okay? So when we're thinking about ions, ions are just charged, right? So they have a positive or a negative charge. It's that simple. There are two kinds of ions. There's what we call a cation, right? And there's an anion. Cations are positive, anions are negative. An easy way to remember that is right in the middle of cation, they put that plus sign for you, right? In the form of that T, so in case you forget which one's positive, you'll always know it's the cation, right? So there you go. This guy, it's called an organic anion. Don't worry about it. It doesn't go anywhere. It always stays inside, okay? It's stuck there. It's stuck because we don't have a channel for it, okay? How many of you have ever been stuck in a room that doesn't have doors or windows? Okay, it doesn't really happen, right? Thankfully. I mean, depends on what kind of friends you have, but that's the way it goes. So, I like to think about this, like a car dealership, right? How many guys have ever been to a car dealership and they have a showroom and they have cars in there, right? And you think, well, how do they drive that car through that little door? That's the job I want, right? Like driving a car through a people-sized door. That seems like so much fun. But they have other bigger doors that open from time to time, right, to allow cars to come in and out. It's just like your cell. Your neurons have special doors that will open from time to time that allow sodium or chloride or potassium to move through that membrane, okay? Now, what opens those special doors, those ion channels? Well, those are neurotransmitters that are going to be released based on other signals and contacts, right? We'll talk about all of that. But we've got these special doors that will open, okay? Now, when we open these doors, we don't really have to force anything, right? So at 6:20, there's essentially a door that opens for this class, and it allows you to leave, right? So right now, I have a high concentration of students in this class. At 6:20, I don't, right? Because the door's open and you guys diffuse out, right? High concentration, you want to go to low concentration, right? Happens every Monday, Wednesday, Friday at 10:50, right? So if you guys take Monday, Wednesday, Friday classes, 10 to 10:50, pretty common class time, right? If you were to check the campus and you were to map the location of students at 10:40, there would be a high concentration of students in all the classroom buildings. Not that many students out on the walkways and out in the other areas of the campus. 10:50 happens, right? Special event. We open up the channels, open up the doors for the students. You guys all diffuse out of the classrooms for a few moments there, 10 minutes or so. You're out. Even distribution of students across campus, and then by 11, you guys are all clumped back up somewhere else, right? Some of you in another classroom, some of you home, wherever it is you go to try to eat an early lunch, whatever it is you do, right? It's the same process, okay? It's going to happen here. When we open a channel, stuff will move through that channel in either direction, but what drives things through that channel is going to be the concentration. So we've got some diffusion, we've got electrostatic pressure, right? That's going to drive this stuff through the channel. So if I open a sodium channel, which way does sodium move? Inside the cell or outside the cell? It's got to move to the inside, right? Because we don't really have any sodium inside right now. So it's got to move inside. We've got a high concentration outside, right? So when I open the sodium channel, sodium comes in. If I open a potassium channel, potassium goes out, right? Because potassium, we've got a high concentration of potassium here on the outside, or on the inside, I'm sorry. So it's going to go out. Now the interesting thing is we have sodium channels, we have special potassium channels, and we have chloride channels. These channels are specific for each ion, right? If you are sodium, you will not fit through a potassium channel. It's not going to happen, okay? If you are a car, you're not going to drive through a people door. It's not going to happen, okay? So don't try it. You get stuck and they turn you away. Okay? So we're going to open specific channels at different times. Again, that's going to be based on neurotransmitters. We'll talk about that later. But when we open those channels, the ions just move according to diffusion, electrostatic pressure, all of these sort of natural sources there. We don't really have to force anything. It just happens, okay? Because those forces are already there. We've already set up this electrochemical gradient, right? We just open a channel and we let it happen. We've already pushed the sled to the top of the hill. All you got to do is let it go down, right? It's that simple. Does that make sense, Kyle? Sounds good to me, too. Now, there's sort of one different way we can move ion channels or ions across the, the membrane. And that's this guy called the sodium-potassium transporter, also called the sodium-potassium pump, also called the sodium-potassium ATPase. For me, I like the word, well, all of them work. I think ATPase gives you some information about why your brain cells use so much energy, right? So you guys know ATP, right? It's kind of that energy token for your cells. Anything that ends in ACE, that's a chopper, right? It just chops things, okay? It's an enzyme, it's going to break things up, right? So an ATPase is going to take ATP, it's going to knock off a potassium, or a phosphate, I'm sorry, and make that an ADP, right? Which is awesome because when that phosphate gets lopped off, it's going to release some chemical energy and it's going to allow you to drive this, this pump or this transporter, right? And what this transporter does is it moves sodium out and it moves potassium in, right? And so we can go back and we can reestablish this electrochemical gradient where we've got a bunch of sodium on the outside and a bunch of potassium on the inside, okay? Because when we open a potassium channel, that was potassium out, we need to get that back in so we can try that process again, right? It's just resetting the process for us later so that we can open these channels and create that same change in memory potential. But this is why, this thing runs all the time. This is why you constantly need energy for your brain, right? This is why you guys love Fun Dip, right? You guys know Fun Dip, just like that straight sugar, the solidified stick, right? It's awesome. Why is it awesome? Because it's glucose. Why is glucose awesome? Your brain runs on glucose, right? And I know how hard you guys study. And I know how much energy you need for your brains because you're overworking it sometimes. And that's why you really want those really high in sugar substances, right? That's why they're so palatable, right? You think, well, why does sugar taste good? It tastes good because your brain needs it, right? That's exactly why it tastes good. And I know that's a little bit of a surface level explanation, but that's it. Why does, I don't know, tree bark, why does that not taste good? Well, you know, your brain doesn't need tree bark, right? So there you go. Not a problem if the sweetest thing you can get your hands on was an apple, right? So that was pretty cool. But when the sweetest thing you can get your hands on is just like, you know, 5-pound bag of sugar, then that can cause you some problems. So you gotta watch out. Hey, check this out. This is the same thing I just told you. Intracellular, you got your organic ions and your potassium. Extracellular, you got your chloride and your sodium. Not a big deal. Here's a nice phospholipid bilayer for you. I didn't want to try to draw that. You didn't want to see my drawing. Right? Here we have that organic anion, can't leave the cell. You've got your potassium, right? A lot of it inside. You got a little bit outside, but mostly inside. You got your chloride and you got your sodium here, okay? Constantly being pushed up against that phospholipid bilayer, but unless we have a channel, right? Unless we've got a channel there for you to get through, you're not going to be passing that phospholipid bilayer. Things that are lipid soluble, they don't carry a charge, right? All of these things we're talking about right here, they have a charge, either a positive charge or a negative charge. They're not lipid soluble. They're not going to slide through that membrane. They need that special channel. Here's your sodium-potassium pump. You pump out 3 sodiums for every 2 potassiums that you pump in. As you're pumping out more positive, what are you creating on the inside? You're creating a negative charge, right? And that's what we want to do. Why? We want to get back to, who remembers that resting membrane potential of negative 65 millivolts? Okay? Once we open all of these ion channels, and I'm getting a little bit ahead of us, but we have to do this to come back. Once we open some of those sodium channels, right, to create an action potential, we've had an influx of positive ions, right? And that's going to take that membrane potential from negative 65, man, we may skyrocket to like positive 40, okay? So that's a massive change, right? Negative 65 to positive 40, right? What happens if you owe the university $6,500, and then all of a sudden the university says, no, in fact, we owe you $4,000, right? That's a big change in your finances all of a sudden, right? Okay, so it's a big change right here from negative 65 to positive 40. But we need to get back down to that negative 65 so later we can kick back up, right? And so we're going to keep running that sodium-potassium pump so we can do that. Constantly going to be running. When we're opening those ion channels, that influx is going to overwhelm this process. That's fine. Temporarily, we do want to overwhelm it so we can have that positive event that'll disappear shortly, right? So we can kind of see that happens. But we want to kind of hang out down there at negative 65 if we can. Questions about this? Every time this happens, you're using up an ATP, right? Okay, so every time you're pumping out 3 and pumping in 2, there goes an ATP. There goes another ATP. Constantly happening. We have a bunch of these in all of your cells, all of your neurons, okay? So they're all over the place. This is one of the reasons, like at any given moment, 25% of your blood flow is going to your brain. Something that only weighs like 2.5% of your body weight is getting 25% of your blood supply at any given moment, right? That's pretty impressive, okay? That's like a really, I mean, how many of you had like a really whiny sibling, right? Who's always getting your parents, yeah, there you go, right? Still bitter about it, I can tell. Your hand went up, and for some reason they got all of the attention, right? The brain is that. That's what the brain is. It's like, give me glucose, give me nutrients, because I said so. Or guess what? I'll make you run out into traffic. And that's exactly what will happen. If you try to starve your brain of oxygen and nutrients, you'll do stupid stuff. I mean, you do enough stupid stuff anyway. So don't, baseline's pretty low. We don't want you going below that, okay? Right, Abby? All right. Let's take 25 seconds to think about that same cartoon. Because we're going to come up on something else here, right? We've been talking about membrane potentials. I've got you set up. You kind of understand some electrical things that are going on. Now we're going to really talk about that really key important event called the action potential, right? But this is the most beautiful, wonderful thing that will ever happen to you. Because if it doesn't happen, you're dead, right? I mean, that's why you know. This is amazing. In fact, every thought, every feeling, everything you've ever experienced can really be just boiled down to a series of action potentials. That's it. That's all it is. Your entire existence is just this. That's it. Just action potentials. If you think you're funny, it's just a bunch of action potentials, right? You and the folks who think you're funny, or at least make you think you're funny, just action potentials. All right, so let's think about this. Okay. First, we have to become really depolarized, right? So we're at negative 65, we've got to start working our way positive. We do that mostly because we're moving that sodium and potassium, right? So we're moving sodium in, we're moving potassium out, okay? As we're doing that, I know you're thinking, well, why am I moving potassium out? If it's positive, don't I want to hang on to it? But we got to set up that negative membrane potential later, so we have to do this, okay? We're going to move it through those ion channels we've already talked about. Not a big deal. Because we've got this big influx of positive, we have this really brief electrical impulse. This whole thing takes about 2 milliseconds, right? That's super fast. Okay? 2 milliseconds. We have this brief electrical impulse. Okay? So normally you're kind of hanging out here around that negative 65. Whoa, all of a sudden you have that brief impulse that's going to go up and you're going to come back down and hang out. Okay? Not a big deal. We do have something called the threshold of excitation. Because we're hanging out at negative 65 and we're fluctuating up and down a little bit, right? Up and down, we're just doing this. Okay? If we just went up just a little bit, if every time we got just one little extra ion of sodium to come in, we fired an action potential, that would be horrible, right? Okay? We would constantly be firing too many action potentials. There wouldn't be any space between them. Your cells would get overexcited, and you would constantly be in a situation essentially like you were having a seizure, right? Because you would constantly be having all of these this overexcitation. So we have to have some threshold of excitation. We've got to have some level where we say, well, you have to at least get up to here before we're going to let you fire an action potential, okay? And that's around 10 millivolts, right? So once we move positive 10 millivolts, right, from wherever we are. If we're negative 65, we get up around negative 55 or so, then we'll fire an action potential. All right. So these guys are ion channels. Don't confuse them with bell peppers, right? I know some of you will. They kind of look like bell peppers, at least in the way they're drawn here. The point is they have multiple segments, right? And these segments kind of wedge in together, okay? Most of the time they're going to be closed. There are some things here you can't see that we'll talk about later, but there are small holes where we will have a neurotransmitter, whatever one's your favorite. It could be dopamine, it could be serotonin, it could be acetylcholine, right? Whatever it is, will fall into that hole, and they'll cause that ion channel to open, right? And when that ion channel opens, these ions will flow through, either inside or out, whichever way it goes, right? Wherever that concentration gradient has things set up for us, okay? It's going to move one way or the other. This is what's going to create that. So if you replace this with sodium, sodium's going to rush in. This guy was at negative 65 millivolts. Now all of a sudden we've got a bunch of positive going in. So this starts slowly creeping up until we get to that negative 55 millivolts, and we're off to the races, right? So we have our action potential. This diagram may be one of the most important in the entire book, right? And especially for this chapter. So this, I would study this. I would memorize this. This is important. You are going to have to write extensively about this particular diagram at some point. When would that be, you think, Casey? On the midterm. Do you have a date when I might have them? I don't know when the midterm is.
I don't know when the midterm is.
It's the only thing you need to know today.
Yeah, like not like those quizzes though.
Casey, minus the calendar.
Not important.
No worries. On your midterm, I would anticipate an essay question about the action potential. I would be prepared to start at the beginning. I mean, this says negative 70, doesn't really matter. Start somewhere and get me to the end of the action potential, right? Again, this takes about 2 milliseconds to happen in your neurons. I mean, it's going to take you about 3 or 4 paragraphs to describe to me, okay? So be ready to do that. You can draw this diagram, label it, tell me what's going on. This is important, okay? I'm trying to tell you now. I'm not going to tell you everything that's on the exam audit, but I'll tell you this, right? So this is going to be there. I think I've offered this class 15 times. Every time I've offered it, this has been an essay question. Okay? I think it's that important. And I tell you, well, I don't care if you know. Trust me, it's easier to grade if you guys know the answers, right? If you kind of know the answers, that's when it gets difficult. All right, so what's going on? We're hanging out at negative 70, negative 65, whatever. We're kind of moving up and down, though. It's not a flat line, right? Because we're getting some of those ion channels to open, and some are sodium channels. Some are chloride channels that are opening, that are letting negative ions come in. When the negative ions come in, then we kind of go down a little bit, right? And so these things, they move in and out, constantly going up and down, up and down, up and down. Now, at some point, we get enough of those sodium channels to open to get us up to that threshold of excitation. Okay? We get enough positive ions inside the cell to change that membrane potential up enough, okay, that we're going to get enough of those to open so that we start to have that major depolarization. Okay? It's going to fly right up, okay, way past 0, all the way up to somewhere around positive 40. Okay. Now on the way up, because remember later we've got to do that sodium-potassium pump, right? So on the way up, as we're going through this process, your potassium channels will actually start to open, and potassium is going to leak out of the cell. Okay? So potassium is going to leak out a little bit. You're going to lose a little bit of positive on the inside. We're going to need that later to reset that membrane potential, right? So we need those potassium ions on the outside. It's going to help us a little bit so we can control the dynamics of this, right? Think about how much faster this would go up and how much steeper it would be if we weren't letting out some of that potassium, right? To sort of temper this a little bit, okay? But we need to do that. When we get to the peak, these sodium channels become refractory. And what we mean by that is the sodium channels won't open, okay? So they're open for a brief period of time, then they're going to close, okay? Now when they close and there's not any sodium coming in, there's still potassium leaving, right? Because potassium is still leaving the cell, guess what happens? We start going back down, so we're going to we're going to hyperpolarize because we become negative, okay? We actually overshoot just a little bit. It's called the after hyperpolarization. So we overshoot that membrane potential just a bit, and then that sodium-potassium pump kicks in, right? Okay, because we're not opening those channels as much anymore, sodium-potassium pump can catch up, and we can get back up to that resting membrane potential, move along until it happens again. Okay? And it just keeps happening over and over again at some specific pattern, depending on the stimulus that's coming in, right? It's not a big deal. Now, when we talk about this refractory period, right, which is kind of interesting, there are actually two refractory periods. There's what we call the absolute, and there's what we call the relative. Okay? For the absolute refractory period, that is a time period when those sodium channels will not open, no matter what you do to them. Okay? They will not open. It's impossible to get them open. They're not going to budge. The relative refractory period, which is a little bit later, is a time period where the sodium channels are difficult to open, but if the stimulation is high enough, if the stimulus is strong enough, those sodium channels will open again, and we can fire another action potential. Now this lets us do a couple things. One of the things is it allows us to create individual spikes, okay? Individual action potentials. If we were coming along and we just go, oh, well, we're positive now, we're just going to stay there as long as there's a stimulus, and we drop back down. That doesn't tell us a whole lot, right? Doesn't give us a lot of information about what's going on in there. Because it could be that pattern, or it could be this pattern. Those are two different patterns, right? They mean two different things. So the pattern and the timing of the action potentials lets you know what information is in there, right? Lets you know what information is being conveyed from one neuron to the other. If it just turns on, and it stays on, and it never goes off, then we don't know. So how many of you are familiar with like Morse code? Anybody? One person? Almost familiar, or were you just adjusting your glasses? Just adjusting your glass, what I thought. So Morse code, right? It's like dots and dashes, right? Okay, anybody in the Boy Scouts? Anybody know a Boy Scout? No? You guys are useless. So it's a series. It's a code, right? It's a series of dots and dashes, right, to represent letters. Okay. This is great. Any of you like ever get lost in the woods? No. Anybody ever been in the woods? All right. 3 people. Yeah, I think if you're wearing flannel, you've been to the woods, right? Okay. Thanks, Bethany. So if you go out in the woods, you might like take a mirror, right? So you can kind of send a signal, right? So you can kind of send a. Hopefully they teach you that, right? And like, don't get lost in the woods school. I don't know if you've been to that or not. You might take a mirror and you might like, SOS or something, right? You guys familiar with that, right? Okay. So it's like a series of dots and dashes that represents letters, right? So the interesting thing is your action potentials sort of work in a similar way. There aren't any dashes, it's just dots, but it's still this pattern that's important, right? And so if you're trying to send someone an SOS message, right, dot, 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 dash, 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 dot, 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 whatever, so that's great. But what if you just like turned on your light and you left it on and then you just turned it off? Well, that doesn't tell somebody what the message is, right? And so they don't know. They're like, well, there's a light. That person must be fine because that's not the SOS signal, right? There's not any information in there. But when you have the dots and the dashes, well, then somebody knows, oh, well, that's a pattern. That means something, and we can go with that. Action potentials are similar. If you just become excited and stay excited the whole time, and there's not these discrete individual peaks the whole time, you don't know what that information is. So this is on for, let's say, 10 milliseconds. Okay. Well, was I looking at an apple or was I looking at a lobster? I don't know because there's not any information in there, right? I was just looking at something for 10 milliseconds. If it's a lobster, well, then I have this pattern of excitation. And if it was an apple, then I have this. Those are two different patterns. They mean different things, right? And so that information is going to be in there. That's why we need these refractory periods to create individual action potentials within that time block of stimulation. Okay? Questions about that?
What did you say the absolute period is for?
Yeah, so the absolute refractory period, they will not open no matter what you do. Okay? No matter how strong the stimulation is, those sodium channels will not open. They will not budge. Okay? Not at all. Relative refractory period is, they will open, but they're not happy about it. Okay? I like to think about absolute and relative refractory periods. This is a perfect time, I think, to talk about, like, football players and so forth, right? If you think about, I think about sort of like offensive linemen, right? And they'll move about like 3 yards, right, per play, and then they take a little break in between as they're kind of trotting up to wherever the ball moves, right? Now, once the whistle is blown until like the ball is put down, that's like an absolute refractory period, right? You cannot run a play during that time, right? Because the referee's moving the ball, right? So it's impossible for you to run a play. Now, relative refractory period is a little different, right? And so, the referee's put the ball down. You have some amount of time you can take, get up to the line, run a play, you can stall a little bit, you can do different things, right? And so I think about, like, absolute refractory period, that's definitely controlled by the referee, but relative, I think about, like, a few years ago, Peyton Manning, one of the best guys at running the 2-minute drill, right? And so I think about the relative refractory period as kind of like last couple minutes of the game, you're trying to squeeze in as many plays as you can because you need to score a touchdown, right? And so you're trying to cut out all of those sort of extraneous time wasters so you can get back up to the line and run another play because the clock's ticking, right? And so you need to kind of fit in more action potentials as you're going. Does that make sense? Great. Don't worry about this too much. There's the rate law. Basically what this says is the stronger the stimulus, the more action potentials it will create. That's what I need you to get out of it. That's not too complicated, right? All right, this is saltatory conduction. Here we are talking about those nodes of Ranvier just a little bit. This has nothing to do with salt. It actually means to leap, right? Okay, so if any of you are like dancers, and I don't mean like, I mean like classical dancers, right? Like ballet sort of dancing, right? Lots of people dance, and that's not always. There's not always a lot of skill involved there, or training, right? Trevor, you may know some of those people, right? So I think that happens. Remember that chart I drew earlier that looks something like this? You could replace athletic prowess with dance over here, like how much you think you can dance, and like your actual dancing skills go down that way, right? I think that happens. So there we go, great charts. So, saltatory conduction, basically what we're going to do is as we go through the myelin sheath, it actually sort of, that action potential sort of decreases a little bit, right? And that's bad, because if that happened forever, it would just kind of disappear. But what we have is those nice little gaps And at those gaps, it gets a little power boost, right? Because we can kind of regenerate it there. It's going to be open. We're going to have ion channels that are going to open, and they're going to boost that action potential forward, right? Which is going to be pretty cool. So you get a little power boost there. It's like on Mario Kart when you run over those yellow wedges, right? You guys are Mario Kart fans, I assume. Right? Is that true? Yeah. It's okay. The cool thing about this is this relates right back to those refractory periods, right? So what if these ion channels could just open and close randomly whenever they wanted and there wasn't a refractory period? Okay? And you're coming along through here, you're just cruising along, right? Cruising along that axon. Now, we want axons to move in one direction, or action potentials to move in one direction only. We want to go from soma to terminal. What happens if you send an action potential and it just makes a U-turn? It doesn't do you any good, does it? You're like, oh, legs, you need to run because, I don't know, the piano's going to fall on me. And then all of a sudden, that signal just goes right back to the cell bodies and, well, comes that piano. What are you going to do about it? It's a bad idea. The refractory period actually prevents this, right? Because once an ion channel, let's say right here, has opened and it's closed, guess what it can't do? It can't open again, right? So that action potential can't go in reverse, it's only got to go forward, right? Because those are the only ion channels that can still open to allow the action potential to move forward, right? Because they can't go backwards. It's brilliant. So as you move along and you go through that refractory period, that absolute refractory period allows the action potential to only move in one direction, in the forward direction. Now, you could artificially, of course, send an action potential or an electrical signal. It wouldn't be an action potential, Brandon, because we're creating it artificially. You could send it the other direction, right? If I was down here and I put in one of those electrodes and I just, sent in, it's a lightning bolt, sent in a little bit of electricity, I could send it back in the other direction. Guess what would happen? It's the same thing, right? It's not going to turn around on me because I still have that refractory period even though I'm really sending it in the wrong way. Sodium channels don't care which way they're in there. They don't care which way the action potential moves. All they do is they open and they close. And they stay closed for a while, and then you can kind of force them open a little bit after that, right? And so that creates this sort of one-way propagation of the action potential. It's pretty cool, actually. Questions about that? Great. All right, we're moving along. So, that's sort of the end of the electrical discussion, right? We've talked potentials, we've talked about moving that action potential, we've talked about how it moves in one direction, we've talked refractory periods, we've talked about how all this fits together. We're letting sodium in and potassium out, and then we're starting that pump again to reset everything, right? So we can try it again in just a few milliseconds, okay? And that's great. Now, the problem is that's all self-contained, right? We're all in this cell, and we're not getting anywhere else, right? We've not talked about how do we get that information to another cell, right? How do we get that information to another neuron, or get that information to your muscles so you can make some kind of activity, right? So let's talk about that a little bit. And this is what we mean when we talk about synapses. Okay? So what do we mean by synaptic transmission? Basically, we are going to take the signal from one cell, one neuron, and get that over to another cell. Okay? For the most part, your neurons are not electrically coupled. There are some exceptions to this, okay? And we'll talk about those exceptions if we need to. But most of the time, they're not electrically coupled, okay? Instead, there's a gap between your cells, and at that point, what we will do is we'll take that electrical signal. Of course, we spent a long time making that an electrical signal. And then we're going to turn it into a chemical signal so we can turn it back into an electrical signal. And I know that may or may not make a lot of sense right now, but hopefully in a bit it will. So we have that electrochemical signaling is what we call it, right? We go from the action potential to neurotransmitters, back to action potential to neurotransmitters. We'll continue this process for however many steps, however many neurons are involved, okay? Now there are a few words we need to know so we can think about this. If we're thinking about the entire setup there between terminals and dendrites and that space in between, we sort of refer to that as the synapse, okay? There's a space in between the terminal and the dendrite or the axon, wherever we're contacting. We call that the synaptic cleft. I'm not going to get too upset if you refer to sort of any or all of this as the synapse, right? A lot of folks do. It's okay as long as you sort of know what we're talking about there and what the difference is. We have two cells involved here. One of these we call presynaptic, right? It's kind of, that's the terminal, that's before the synapse. That's the guy sending the signal, right? And then we have something called postsynaptic, okay? That's the person receiving the signal, okay? That's the cell receiving, right? Not too big of a deal. We do have a phrase called postsynaptic potential. There's that word potential again, so we're thinking about electrical signals, right? We're not thinking about a full-blown action potential here, right? We're just moving closer to or farther away from that, either with a depolarization or a hyperpolarization. We have things called excitatory postsynaptic potentials. That's an EPSP. And then if it's inhibitory, that's an IPSP, right? So did we make this cell more likely to fire an action potential or less likely to fire an action potential? That's really all we're thinking about there. Did we excite it or did we inhibit it? We're not making it do an action potential because we've got to get a bunch of other inputs involved to do that. But are we pushing it closer to or farther away from that action? All right. And then we have something called a binding site. This is very, very complicated. This is the site where the neurotransmitter binds. I know, right? Nobody was ready for that. So we have a receptor. It is a protein. It's not a big deal. And we have a thing called a ligand. Don't worry about that. A ligand is, for our purposes, is just a neurotransmitter. It's just a substance, a thing that sort of fits in to that receptor and will cause that receptor to open. Lots of folks use the lock and key analogy for that, right? Works fairly well, despite it's a little hack if you ask me, but you know, people use what tools they have, right? So there you go. All right, we've already talked about the dendritic spines, not a big deal. These actually come off of the dendrites, so you have your dendrites, and you have little things that stick off of that. So now it's time to talk about the second key to transcendental enlightenment. It's surface area to volume ratio, right? If you understand surface area to volume ratios, life makes a lot of sense, right? Why are red blood cells shaped the way they are? It maximizes their surface area without creating too much volume, right? Because the problem is if it was a sphere, right, if you think about red blood cells, and you were an oxygen molecule in the middle, do you know how long it would take you to like travel to the surface so that you could like unload out of the tissue? It takes too long, right? It's just not reasonable. If you're donut shaped, wow. I mean, man, that just really maximizes your surface area without maximizing your volume, right? And so that those oxygen molecules can jump on and off as needed. So if we think about that and think, well, surface area is important. If you think about the structure of your cortex, right? Why is that guy crumpled up? If you were to take the cortex off of your brain and you were to spread that out, it's about 3 feet by 3 feet, right? Remember that discussion we had about your mother's hips? She's not letting something 3 feet by 3 feet go through there, I promise you. If all of a sudden scientists said, hey, all babies born in the future are going to have heads that are 3 feet by 3 feet, the human race would cease to exist by whenever you guys die. Because nobody's going to be having kids. It's not going to work. So, surface area to volume, why does that apply here? Dendritic spines are a way to increase the surface area of the dendrites without increasing the volume. They increase the number of places where those other terminals can make contacts, right? So we're increasing the surface area a little bit so we can cram in those 10,000 contacts without making each neuron too big, right? So surface area to volume ratios. Very important. Not a big deal. Okay, so dendritic spines, talked about that. Presynaptic membrane, that's just the guy dumping out the neurotransmitter. Postsynaptic membrane, this is where we're going to have that receptor in that ion channel. It's going to receive the message. Not a big deal. Here's the synapse. Here is the terminal coming down. These are things called synaptic vesicles, right? They are jammed full of neurotransmitters and other things that might be needed at the synapse, probably coming down from the soma, coming through that axoplasmic transport that we talked about, right in those microtubules. They'll kind of hang out down here in this terminal until we get that electrical signal to come through. Once that electrical signal comes through, these guys get undocked and they can float down to that presynaptic membrane. They merge with the presynaptic membrane. They're just a big sphere. When they merge, they just crack open and they dump out the neurotransmitter right here at the synapse, right at that cleft. Those neurotransmitters just diffuse across that synaptic cleft, and then they will bind with the appropriate receptors on the postsynaptic membrane. When they bind to those receptors, ion channels open. We've already talked about that whole story, right? Sodium comes in, or if you're thinking about chloride, so kind of closes things down. Assuming sodium comes in, and we just fire an action potential and we continue the process. Questions about that? Everybody understand the basic idea? It's not too bad. There are different kinds of receptors, and we should think about these. Talk about these different kinds of receptors. One of these are inotropic. Okay. This is basically the story I've been telling you now, right? Okay. This is the kind of the straightforward story. You've got an ion channel. It's closed. It has a binding site here. Whatever that neurotransmitter is comes in and it binds. Once it binds to the binding site, it causes a change in the shape of the ion channel. So in this case, it opens up, right? Wedges that open, and your ions flow. In this case, it's showing inside. It could flow out of the cell as well. Sort of depends on whatever the channel is, and again, that ion concentration. Cool thing about inotropic receptors, they're direct. It's a one-to-one relationship, right? You get one molecule, you open one channel, right? Lots of ions can come through, but we're opening one channel. It's sort of spatially tied, right? It's spatially coupled. That's a good word. So what I mean by that is the receptor and the channel are kind of all one integrated unit, right? Okay. It all happens right there. This is also fast. There's no delay. When the ion channel, or when the receptor binds with that neurotransmitter molecule, it opens. It's that simple. There's not really any delay. It's right there. Okay. There's a second type of receptor, right? Metabotropic. These guys are a little different. Okay. Here's the receptor. The molecule binds to the receptor. That neurotransmitter binds to it, but it doesn't directly open an ion channel at that time. It starts a second messenger system. It involves G proteins and second messengers. Some of you guys may have heard of these things. If you haven't, don't worry about it too much. We're not going to hit you too hard. Basically what happens, the molecule of neurotransmitter binds to the receptor, and that causes the second messenger system to activate, right? And so what happens is that receptor says, okay guys, go out and open ion channels. So instead of it being direct, it's sort of indirect. It's more of a one-to-many situation, right? We're not like a cascade. We're not doing a one-to-one. Notice here we're showing, this guy's opening 5 different ion channels, right? So we've activated one metabotropic receptor, but we've opened 5 ion channels. There's a little bit of a time delay here, right? Because not only do we have to have that molecule bind to the receptor, but then we've got to send out that second messenger system, right? We've got to activate those G proteins and such, and I'm not talking like 10 minutes. There's a little bit of a time delay here, so it takes just a little bit longer than those inotropic receptors. We are also dealing with a situation that is spatially diffuse, right? And what I mean by that is, here's the receptor. The ion channels are located elsewhere, right? That second messenger could really just travel wherever, okay? Now we're not going to send it too far, obviously, because that doesn't make sense for sort of this rapid action potential business. But there is going to be some distance between the receptor and that ion channel that it eventually opens, or a number of ion channels that it eventually opens. For all practical purposes, though, it still makes an action potential, right? It just does it in a slightly different way. Just a little bit of a wrinkle on the story, something to keep in mind. Later, we'll talk about specific substances that will activate inotropic or metabotropic receptors for different ions and for different substances, okay? And some of that may be important. We'll talk about it when it is. If it's not, you just know it. So don't worry. Synaptic vesicle, we talked about that. Not a big deal. Release zone, that's just, again, zone of release. We already talked about excitatory postsynaptic potentials and inhibitory postsynaptic potentials. There are two things we do want to talk about, though, because this is a discrete, short-lived process, right? We don't want action potentials to go on forever, or again, we lose all of that information. Quick action potentials, okay? We want that to happen, we want it to stop. There are two basic ways that we can end the action of neurotransmitters, right? One of those is a process called reuptake. When you dump out neurotransmitter into the synapse, okay, reuptake works by sucking that neurotransmitter back into the cell that released it, okay? So some of those cells that release neurotransmitter have other proteins hanging off of them with little arms that reach out, literally, and when they latch onto a molecule of the neurotransmitter, they'll just pull it right back in. We'll recycle that, we'll use it again, and they just keep doing that process. We already briefly mentioned SSRIs. I just bring those up, most folks are familiar with those. That's a selective serotonin reuptake inhibitor, okay? So when you take an SSRI, what this does is it prevents that reuptake process, okay? Why would you take an SSRI? You need to increase the amount of serotonin you have, right? So we're going to increase the serotonin that's available at your synapse. We'll talk a lot about that kind of stuff later. But we're going to do it in this case by blocking that reuptake process so you have more serotonin there available for you to use, okay? The other thing we can do is enzymatic deactivation. So basically what happens here is we have enzymes that break down neurotransmitters. Okay? One of those is acetylcholinesterase. This guy breaks down acetylcholine. Acetylcholine is important for a lot of things. One of the things it's important for is muscle contractions, right? I don't know if anybody's ever contracted a muscle before. That's not the same as contracting a disease. So it's a slightly different process. So when you contract a muscle, that's awesome. You're using a bunch of acetylcholine. However, if you want that muscle to not be contracted, you need to break down that acetylcholine so you can relax that muscle, right? You can imagine if we didn't have acetylcholinesterase and you had like one time you could flex, you better make sure it's a good pose because you're going to get stuck there, right? And so you really need to have that acetylcholinesterase so you can break down acetylcholine and you can relax your muscles. It's used in other places too. Acetylcholine is also for cortical vigilance. We'll talk about that later. But enzymatic deactivation. What's another sort of another antidepressant that a lot of folks have used in the past? They don't prescribe this as often now, this class of drugs, because the side effects and the dietary restrictions are kind of steep. It's MAOI, it's monoamine oxidase inhibitors. We said anything that ends in an ACE is an enzyme that chops stuff up, right? So monoamine oxidase typically destroys your monoamines. That includes dopamine, for example. If you take an MAOI, what happens is instead of breaking down dopamine, you have more dopamine available, right? It's just going to be there. We're going to prevent that enzymatic deactivation step. Problem with MAOIs is you can't have wine or cheese, right? So I know that's a real big detriment to a lot of folks, right? I don't know. There are a lot of other dietary restrictions as well with MAOIs, particularly like fermented foods. That's why wine and cheese are on the list. It'll really interfere with that. Lots of other drugs interfere with MAOIs as well. If you're on an MAOI, I assume you know that. If you don't know if it's an MAOI, check, because like everything else you take says, like you pick up a bottle of Pepto and it says, well, don't take if you're on an MAOI, right? So you really have to be careful about that. That's why they would rather prescribe an SSRI, because they'll get typically the same results. Some folks are resistant to SSRIs, and you have to jump down to some of these other drugs that have different side effect profiles. But that's a conversation for a couple of weeks. Questions about this? No? All right. This is basically showing you inotropic receptors, and then here's a metabotropic receptor. This time we're talking about a different ion, so we want to kind of add a wrinkle. That's calcium. Calcium is an important ion that we'll talk about as well with learning and memory and some other things later. But it activates that second messenger system with the enzyme and those sort of downstream effects, right? All right, questions about that? All right. Here's the reuptake process. Here's one of those transporters, kind of a generic drawing. We have this hump here was a synaptic vesicle that just merged with that membrane, right? Not only can we dump out neurotransmitters, but let's imagine I needed a new transporter, right? Let's say some of those transporters are damaged and I wanted to send new transporters down. I could build those transporters in to this synaptic vesicle, right? That'd be cool. And then when I merge it with that presynaptic membrane, I've already embedded my transporters right there. So anytime we need to put in new proteins in the membrane, if we need to insert proteins in the membrane, we can do that through that synaptic vesicle system. It's not just for releasing neurotransmitters, so it kind of has that secondary function there as well. Sorry, I think this screen's about to go out. It keeps like changing colors on me and I can't see it. It's that or I'm having a stroke, and I don't think it's the first time. So one of the two, we'll figure it out eventually, right? I was going to say if what I'm saying doesn't make sense, then maybe, but that probably didn't help. Let's see. Neural integration. I mean, geez, we've been talking about this all day, really, right? So right now, let's just have a slide on it. Basically, what we're doing is you're taking any of the excitatory postsynaptic potentials and adding those to the inhibitory postsynaptic potentials, right? To see if we get to that threshold of excitation, okay? It's pretty cool, right? I like to think about this like if you guys, anybody make a big decision lately? I don't have to share what that is. Let's imagine you were going to buy a car, right? That's a big decision. And there are a lot of factors that go into buying a car, right? The price of the car, what your budget is, reliability. Maybe you ask a bunch of people, right? Like, hey, I'm thinking about buying this car. What do you think about it? Oh, well, I've had trouble with that kind of car. This kind of car is good. And you get all of these votes and you integrate that into some decision, right? And then some of you just go off and buy something stupid anyway. Your neuron's not going to do that. It's going to weigh these positive and these negative postsynaptic potentials, right? These IPSPs and EPSPs. And then it's going to integrate that. And it's going to come to some sort of conclusion there, and it's going to fire an action potential or not. Kind of depends on the result of that math and that calculation, right? This is another one of those times, Kyle, where we can take that three-step process. Input, calculation, output, right? Here are all of the inputs coming in. This neural integration is the calculation, and the output is action potential or no action potential. It's pretty cool. We should talk about autoreceptors too. How many of you have ever done something and then it was like really important that you realized that you did it? Yeah, that happens more than you think. So an autoreceptor is basically a receptor on that presynaptic neuron. So this is the guy releasing neurotransmitter. And what it does is it actually is sensitive to the neurotransmitter that it's releasing. So if this is a cell that releases dopamine, we're going to have dopamine autoreceptors, and they're going to be basically measuring the amount of dopamine that you're releasing. And when you've released enough dopamine, those cells will get activated and they'll shut down release. That's pretty cool, right? I mean, I think it's pretty cool. So that way you know, hey, I've done enough of this. I don't need to do any more. Let's not waste any more dopamine. We've reached the max of what we need to do. And it's definitely for regulating that internal process. Now, in a couple of weeks, we're going to talk about some cool things you can do with your autoreceptors, right? Because you can stimulate these guys or you can block these guys. So if you're thinking about this for a minute, what happens if I stimulate an autoreceptor? Well, then I think I've released a bunch of dopamine or acetylcholine or whatever, and I stop releasing it, right? So it actually shuts down the response. If I block the autoreceptor, doesn't matter how much of the neurotransmitter I dump out, I never realize I've done it. So I just keep dumping out dopamine or dumping out serotonin. I just keep doing that, right? And so there are drugs that you can use to block these autoreceptors or stimulate autoreceptors to change the effects of the neuron. We've already talked about these other types of contacts, so no worries there. Nothing to get too excited about. This is basically what we call axo-axonic terminal contacts, another terminal. That's not typical, but it does happen. There are other things that will, other than neurotransmitters, we should talk about briefly, right? Other things that will affect your communication there. Some of these are called neuromodulators. These are guys that by themselves will not cause an action potential, but they will help an action potential happen or not happen, right? And so they'll kind of alter the effects. Typically these guys are what we call peptides, so they're going to be sort of short segments of proteins. We also have hormones. We will talk a lot about hormones when we get to some other chapters. Later we've got chapters on emotion, which will cover aggression, and reproductive behavior as well. Obviously hormones are quite substantially involved there. And so hormones can, particularly steroid hormones, can affect behavior. Questions about that? And then there's that.